Within every living cell lies a molecular blueprint that dictates its form, function, and destiny. This blueprint contains a complex code that has evolved over billions of years to create the diversity of life we see today. This blueprint, known as deoxyribonucleic acid, DNA, carries the genetic code, a sequence of instructions that determines traits ranging from eye color to susceptibility to diseases. DNA is a double helix, composed of 4 chemical bases: adenine, thymine, cytosine, and guanine. These nucleotide base pairs form the rungs of the DNA ladder, encoding the information that makes each organism unique. For millennia, the structure of the DNA code remained unknown. It wasn't until the 20th century that we began to fully unlock the secrets of DNA. Advances in technology and a deeper understanding of genetics have given us unprecedented control over the code of life. Scientists had finally developed the tools to edit an organism's DNA code. These revolutionary gene editing technologies allow for precise modifications to DNA, opening up possibilities that were once the realm of science fiction. Gene editing technology holds the promise of curing diseases, enhancing crop yields, and even resurrecting extinct species. Imagine a world where genetic disorders are a thing of the past, and where food scarcity is addressed through engineered crops. Yet, with such technological potential comes important ethical considerations regarding the ability to genetically engineer organisms, the potential for unintended consequences, and the implications on human equality. Gene editing, in its simplest form, is the process of making precise changes to the DNA sequence of an organism. Imagine DNA as a long chain of letters, and each gene as a specific word within this chain. Gene editing allows scientists to specifically target and modify these words, thereby altering the instructions they carry. In the case of DNA, these letters and words are made from the sequence of adenine, thymine, cytosine, and guanine nucleotides. The DNA sequence. The exact sequence of A, T, C, and G determines the genetic code. Mutations in this genetic code can result in a faulty gene that causes a particular disease. Gene editing allows scientists to correct misspelled words or to insert a new word. In other words, to correct abnormal DNA sequences that may cause a disease. By replacing the abnormal DNA sequence with a healthy DNA sequence, gene editing can correct faulty genes. Alternatively, gene editing allows scientists to delete harmful words altogether. In other words, it can specifically target abnormal genes and disrupt their DNA sequence, effectively resulting in gene deletion. Gene editing, therefore, has the potential to prevent the expression of faulty genes from causing unwanted disease traits. The ability to precisely manipulate the sequence of DNA has opened up a world of possibilities, transforming medicine, agriculture, biotechnology, and environmental science. Although the concept of gene editing has been around for decades, its widespread application was hindered by the limitations of early technologies. These early methods, while groundbreaking at the time, were cumbersome and lacked precision. These methods were often inefficient, expensive, and prone to errors. Scientists faced numerous challenges, including high costs and the frequent occurrence of unintended mutations or off-target effects, which made the process unreliable and difficult to scale. However, the discovery of CRISPR in 2012 revolutionized the field of gene editing. This breakthrough provided a more accurate, cost-effective, and efficient method for modifying genes, opening up new possibilities for research and treatment. CRISPR, short for clustered, regularly interspaced short palindromic repeats, is a bacterial defense system that has been repurposed into a powerful gene editing tool. This system, known fully as CRISPR-Cas9, was originally discovered in bacteria and has been adapted to target and modify specific genes in a wide range of organisms. When introduced into a cell, the CRISPR-Cas9 is guided to its target gene within the nucleus. The CRISPR-Cas9 contains a short RNA molecule known as the guide RNA. This allows scientists to design CRISPR-Cas9 that precisely targets specific sequences within the target genome. Careful design of guide RNA sequences allows CRISPR-Cas9 technology to edit only the desired gene that requires correction. The Cas9 is a DNA endonuclease enzyme that acts like a pair of molecular scissors. Cas9 precisely cuts DNA at a specific location, which is directed by the guide RNA. This precision allows scientists to make targeted changes to the genome, reducing the risk of off-target effects and increasing the reliability of the edits. Once the DNA is cut, the cell's natural DNA repair mechanisms kick in, allowing scientists to either disrupt the target gene or introduce desired changes. This ability to harness the cell's own repair processes has made CRISPR-Cas9 an invaluable tool in genetic research, with applications ranging from agriculture to medicine and beyond. While CRISPR-Cas9 has taken center stage in recent years, it is not the only gene editing tool at our disposal. Other techniques, such as TALENs, transcription activator-like effector nucleases, and zinc finger nucleases, were developed earlier and continue to play a role in research and clinical trials. TALENs and zinc finger nucleases also function as molecular scissors, but they rely on different mechanisms to recognize and bind to specific DNA sequences. Despite their differences, all these techniques share the same goal: to enable precise and efficient modifications of the genetic code. One of the most promising applications of gene editing lies in the realm of medicine. Scientists envision a future where genetic diseases, such as cystic fibrosis, Huntington's disease, and polycystic kidney disease, could be cured by correcting the underlying gene defects. CRISPR-Cas9 has already shown promise in clinical trials for treating blood disorders like sickle cell anemia and beta thalassemia. In these trials, stem cells were extracted from the patient's own blood, genetically edited to correct the faulty gene, and then transplanted back into their body. The results have been encouraging, with some patients showing significant improvement or even complete remission of their disease. Beyond treating diseases, gene editing holds immense potential for revolutionizing agriculture and addressing global food security challenges. By modifying the genes of crops, scientists can enhance yields, improve nutritional content, and confer resistance to pests, diseases, and harsh environmental conditions. Imagine tomatoes with a longer shelf life, rice with increased vitamin A content, or wheat that can withstand drought conditions. These are just a few examples of how gene editing could contribute to a more sustainable and equitable food system. The applications of gene editing extend beyond human health and agriculture, reaching into the realm of environmental conservation. Scientists are exploring the use of gene editing to protect endangered species, control invasive species, and restore damaged ecosystems. For instance, researchers are working on developing gene-edited mosquitoes that are resistant to malaria, which could help curb the spread of this deadly disease. In another example, scientists are investigating the use of gene editing to control invasive species that threaten native biodiversity. While the potential benefits of gene editing are vast and inspiring, the technology also raises profound ethical concerns. One of the most pressing issues is the potential for unintended consequences. Altering the genetic code of an organism can have ripple effects throughout its biology, leading to unforeseen and potentially harmful outcomes. For example, a gene edit intended to enhance a crop's resistance to pests could inadvertently make it more susceptible to another disease. It is crucial that we proceed with caution, conducting thorough research and risk assessments before widespread deployment of gene-edited organisms. Another major ethical concern is the potential for gene editing to exacerbate existing social inequalities. Access to gene editing technologies is likely to be unevenly distributed, with wealthier individuals and nations having a significant advantage. This could lead to a scenario where the benefits of gene editing are enjoyed by a select few, while the rest of the world is left behind. Ensuring equitable access to gene editing technologies and addressing the potential for genetic discrimination will be crucial for realizing the full societal benefits of this powerful tool. Gene editing is a powerful tool with the potential to revolutionize multiple aspects of our lives, from curing diseases to ensuring food security and protecting our environment. This technology allows us to make precise changes to the DNA of organisms, opening up new avenues in research, medicine, agriculture, and conservation. In medicine, CRISPR gene editing holds the promise of curing genetic disorders and even eradicating certain diseases. In agriculture, it can lead to more resistant crops, ensuring a stable food supply for the growing global population. In environmental conservation, gene editing can help protect endangered species and restore ecosystems. However, this revolutionary technology also comes with certain ethical considerations. Altering the genetic makeup of living organisms raises questions about the long-term impacts and potential unintended consequences. It is our collective responsibility to ensure that gene editing is used responsibly, equitably, and for the benefit of all humankind. This means establishing robust ethical guidelines and regulatory frameworks to govern its use. By harnessing the power of gene editing responsibly, we can create a better, healthier, and more sustainable world for generations to come. I hope you enjoyed watching this video from Science Curious. Don't forget to like and hit the subscribe button. Thank you.